Your career begins here at Satyabama Institute of Science and Technology, a deemed to be university, a nationally recognized and globally acclaimed destination to accomplish your academic aspirations. The Colonel Dr. JPR Research Hub, along with the International Research Center, houses advanced research facilities that have 13 centers of excellence, 10 plus technology transfers, 100 plus patents, 10,000 plus reputed publications. The Satyabama Center for Advanced Studies is a state-of-the-art facility built with the aim to cater to the futuristic aspirations of our students with the aid of a metaverse studio, drone labs, maker's space, media center, advanced characterization facilities, ICT-enabled classrooms, and supercomputing laboratories. The engineering departments are equipped with sophisticated infrastructure, fulfilling the academic and hands-on requirements of our students. The architecture department is furnished with sophisticated design studios and spacious drawing halls, all of which support the learning process of our postmodern generation architects. The Satyabama General Hospital, along with the School of Dental, Nursing, Pharmacy and Physiotherapy, have advanced clinical facilities. The monumental Central Library houses a diverse collection of books, periodicals and journals. Additionally, it also has an open-air theatre with a seating capacity of 1,000. The Management, Arts, Science, Law and Hotel Management departments comprise of all the necessary and required facilities, creating the perfect ambience for our far-sighted students and catering to their skill set needs with the end goal of equipping them to live their dream. Our chain of laboratories with practical exposure equips our students to build a successful career. Our institution has a team of dynamic and outstanding teachers who are ever reachable to our enthusiastic students, promoting the spaces to learn and places to grow. Greenery around the campus greets and accommodates our students, who are the backbone of a developed nation, edging them on to achieve the summits of their success. Satyabama Institute of Science and Technology.
Uh, Good morning, everyone. It gives me an immense pleasure to welcome you all to another insightful session of the Ocean Research Knowledge Series 2026, organized by the Center for Ocean Research and National Facility for Coastal and Marine Research, Satyabama Institute of Science and Technology, with the support of Ministry of Earth Sciences, Government of India. ORKS 2026 is a scientific lecture series aimed at bringing together researchers, students, and ocean research enthusiasts to gain valuable insights from experts working in diverse areas of marine and ocean research. A very warm welcome to all the participants joining us today. Today, we are pleased to have an interesting and highly relevant expert lecture on genomic techniques in marine science. Today's session will provide us with valuable insights into the application of genomic techniques in understanding the marine environment. It is my privilege to introduce our today's speaker, Mr. Raghavendra Kumar. He's an assistant professor research at Center for Ocean Research, Satyabama Institute of Science and Technology, Chennai. We are delighted to have him with us today to share his expertise and insights on genomic techniques and their applications in marine science. We warmly welcome you, sir. Without taking much time, now I invite Mr. Raghavendra Kumar, sir, to take over the session and enlighten us on genomic techniques in marine science. Over to you, sir.
Yes, thank you so much. Sumesh, is my video is like visible?
Yes, sir. Yes, sir. Yes, sir. This is visible, sir.
I cannot see this thing, but okay. Uh, Good morning, everyone. I'm Raghavendra Kumar, Assistant Professor Research, National Facility for Coastal and Marine Research, Satyabama Institute of Science and Technology. And today's my topic is in genomic techniques in marine science. Yeah. So yeah, this is our facility, our genomic facility in NFCMR. Here we have Limnanestic 2000, Promega Spectrum Compact Series System Sanger Sequencer, Digital PCR, Portable Nanopore Sequencer. Then, yeah, okay, let's start with the session. And here, okay, my topic is in marine genomics. So, over Earth's surface, our 70% of the Earth's surface is covered with ocean. Still, some of the organisms we are not able to identify. What are the organisms present in marine ecosystem that we have not identified yet? So here comes the genomics techniques. With this genomic techniques, we can identify what are the organisms present in the marine, and it will help in biodiversity analysis and ecology of life in marine environment. So marine genomics is a study of organism at DNA level in marine genomics is a field where we can study organism at DNA level, which helps in identifying organism. So one important advantage is that some genomic approaches, not all the genomic approaches, some genomic approaches can be non-invasive. For example, we can take water and identify what are the organisms present in the particular environment. So this marine genomics support conservation, sustainable use of marine resources. And yeah, so genomic give us information that traditional observation method cannot provide. Now we have moved to genome level understanding of marine organism. So evolution of genomic techniques. So genomic field is growing rapidly. So initially we have identified species using physical characterization. Now then it comes PCR and Sanger sequencer where the species can be identified at DNA level. A barcoding method was used for identification of the species. Next, it is NGS. In NGS, we can study the entire genome of a particular organism, and simultaneously we can run millions of DNAs. So now we are in multiomics and AI. So now, as I told, we are moving towards we are moving from this field is moving from just observing the physical characterization towards the genetic level understanding. So we are using genome like DNA for understanding the marine organism. So DNA sequencing technologies. So we have three different types of sequencing. First one is that is called a first generation sequencer, which is Sanger sequencing, and then NGS, next generation sequencing, and then third generation sequencing. So Sanger sequencing, this is called as first generation sequencing, which is used for sequencing small DNA fragment. Yeah, small DNA fragment or any particular region or particular gene. So how it comes under genome technique? Like, yeah, now we are using this for sequencing small areas, small regions like small genes like that. But initially, we have used this Sanger sequencing for Sanger sequencer for studying the complete genome of human, that is human genome for that. So we can consider this Sanger sequencer under genomic technique. Yeah, we can study genome also. Then next generation sequencing. This is a high throughput sequencer which we are using is Illumina. Though this is short read fragments, and with this we can sequence the entire genome of any organism. We can sequence large animals from fish to shark, or as well as microorganism also. Then we have a third generation sequencer, which is long read sequencer, platforms like PacBio and Oxford Nanopore Technologies. So yeah, this is the simple workflow. Initially, we have to how the genomic techniques will work. Like initially, we have to collect samples, then followed by DNA isolation and DNA quality check. This is followed by DNA barcoding. DNA barcoding is identification of the particular organism using Sanger sequencer. If we want to study like in depth of a particular organism, we can use this next generation sequencing, where we can do a whole genome sequencing which map the complete genetic architecture of the marine organism. So yeah, DNA isolation. So DNA isolation, why I'm talking about DNA isolation in particular? Because DNA isolation is a crucial step for any genomic technique. We should able to get a very high quality DNA. This will help in successful outcome of the downstream process, whether it can be Sanger sequencing or NGS. For both the techniques, whatever the genomic techniques, we have to get a high quality DNA. So what are the key steps involved in DNA extraction? First one is sample collection. Sample collection is important one, where we have to transport the sample collection must be in a sterile condition. Okay. Then we have to do a shipment of sample in cold condition, like ice pack is enough for DNA. If we are going to work on RNA, we have to do a dry ice shipment. and we have to ship that sample to the lab as early as possible within a 24 hours or 2 days. Make it sure it should be shipped to the lab in 24 hours. Then followed by the sample collection is the DNA isolation. So DNA isolation can be done by either CTAB method, that is the conventional method, or isolation kit. Many isolation kits are available in the market like Kaizen, Zymo, and Ambion. We can use those kits for particular samples. For example, there are kit available for tissue DNA isolation. There are kit available for plant tissue DNA, plant DNA isolation, and soil DNA isolation. But if we can also use CTAB method also. This is a conventional method, but it can be, we have to, we should have more care when doing this CTAB based conventional method. Okay. Initially, the DNA isolation step will have cell lysis. Cell lysis means it will lyse the cells and it will help in protein degradation, and the DNA will lyse out of the cells. Then it is followed by removal of contaminants and DNA purification. So removal of contaminants means so any salt or other any proteins get
Sir, your voice is not audible, sir. And PPT is also not visible, sir.
so means it now it is working. Hello.
Voice is audible, sir, but PPT is not visible, sir.
Yeah, let me start. I think it connection got cut.
Okay, sir. Okay.
Okay, 1 second. Let me know this thing. And also where it
Now it's visible, sir.
where it stopped.
The previous slide, sir. Yeah. Yeah, from
Which step, which PPT, which slide it is stopped?
yes, sir. Yes, sir. You can continue, sir.
This one or
Yes, sir. This one.
yeah. Okay. So sorry. And yeah, again I will repeat this thing. So initially it has like DNA sequencing technologies have three different this is the workflow of DNA sequencing technology. First, we have to do a sampling, that is sample collection, followed by DNA isolation and QC. Then either we have to go for DNA barcoding. First, yeah, we will go for DNA barcoding. Here we have to identify the specific organism using Sanger sequencer. And then if we need a in-depth idea on the particular organism, we have to go for NGS, which we can use a whole genome sequencing to map the complete genetic architecture of the marine organism. So yeah, now the DNA extraction process. So DNA extraction is the crucial one in any genomic techniques. So yeah, we have to get a very high quality DNA, which will help in successful outcome of the our downstream process. Either it can be a Sanger sequencer or NGS. Any genomic technique need a high quality DNA. So what are the key steps involved in the extraction of high quality DNA? So what is high quality DNA? Like we should able to get a good concentration, enough concentration to process the downstream analysis, and then your DNA should be intact DNA, and we have to take care of the purity. So what are the key steps involved in isolating high quality DNA? One is a start with sample collection. So sample collection is the important step in isolating DNA. We have to collect the sample in a sterile condition, then followed by the shipment of the sample. Sample shipment should be done in a cool condition, that is like ice pack shipment or dry ice. In case of DNA, we can use a ice pack, and in case of RNA, we can use a dry ice. Okay. This shipment should be happen within a 24 to 48 hours. This should reach the lab within 24 to 48 hours. There should not be any delay, which may affect the quality of DNA. Once we receive the sample, once the sample enters the lab, that we have to do a DNA isolation. DNA isolation has some important steps like cell lysis, removal of contamination, DNA purification, and quality assessment. So we can use CTAB method, which is the conventional method, or any commercially available kit-based method. Like there are a lot of commercially available kits in the market, which is Kaizen and Zymo and Ambion. So these kits are available for individual samples, like specific to any samples. Like we have a tissue DNA isolation kit, and we have a plant DNA isolation kit, and we have a soil DNA isolation kit. So alternately, we can do it in a CTAB also. So here cell lysis is the first step. Cell lysis means it will lyse the cells and it helps in protein degradation, by which DNA can be lysed out of the cells. Then it is followed by removal of contamination and DNA purification. So what are the like removal of contaminants means? What are the contaminants will be there? Like it can be any salt or any proteins, polysaccharides, like that. So this can be removed by purification step. Like in kit, we use a spin column method with spin column plus wash buffer, which is used for purification. And in conventional method, we use as ethanol with wash the contaminant. And then when we are using kit, it is we have a spin column which will help us in getting a high quality DNA. But in conventional method, I would recommend for a magnetic bead based purification. Okay. Once the purification is completed, we have to do a DNA elution with TE buffer. Once DNA isolation is done, what we have to do next? Next is doing quality check. That is quality assessment of the DNA, which can be done by. So first, what is high quality DNA? High quality DNA means we should able to get a highly concentrated DNA. Like we should get enough concentration to process to the downstream analysis, and then DNA purity is must, and then we should get an intact DNA. Okay, so first what we do, there are three different methods we can use for quality assessment. One is nanodrop spectrometer, qubit fluorometer, and agarose gel electrophoresis. In nanodrop, we measure concentration of the DNA. Then purity. Purity is by using 260 bar 230 and 260 bar 280. So both should be in a range between 1.8 to 2. Then followed by agarose gel electrophoresis. We have to load the DNA. We have to run it in gel. which shows our integrity of the DNA. Like we should not get any square and fragment in our DNA. Okay. Then we can use qubit also for measuring the concentration. In this, qubit will use a fluorescent dye which will bind to the double-stranded DNA and give us the concentration. So this concentration can be more accurate. But it doesn't tell anything about the purity of the DNA. So we have to use nanodrop again. Then once the DNA isolation is completed, either we have to go for a Sanger sequencing or NGS sequencing. So Sanger sequencing, as I told earlier, it will do a sequencing of any particular region or gene. It's like we can sequence 900 base pair at a time. So as usual, the workflow is like this, like sample collection will be there, followed by the DNA isolation. Then we have to do a PCR amplification of our particular target gene or gene of interest. It can be anything like a gene to identify a particular organism or our gene of interest to study the how the genes is present in the particular organism. Then once the PCR, once the product, once our gene of interest product is amplified, we can go for a sequencing PCR in which we add DDNTP, that is dideoxynucleotide triphosphate. This DDNTP will assign different different colors to the individual nucleotide. For example, A will emit green color and T will emit red and G blue, C black. So in sequencing, when we load the samples in sequencing, this DDNTP will emit different colors fluorescence, which will be captured by the instrument and that will come out as a raw data. So once we got the raw data, this raw data will be converted to the human readable format called as like base calling. There are inbuilt software. Each instrument will give their own inbuilt software which convert the raw data to the human readable format, that is called as base calling. So once we got that base calling, what is the format is like FASTA format. Once we get the DNA sequence, we can just do the quantic alignment, like using bioedit or mega. We can do the forward and we can use the reverse forward and the reverse sequence for making a quantic. This quantic can be submitted to the BLAST to identify the similar organism or the gene. Similar organism or any gene of interest. This BLAST will give us the our similar organism, how it will give, like it will give using percentage coverage and percentage similarity, coverage of the particular gene and by the identity of the particular sequence. Then NGS will follow the same protocol like sample collection, DNA extraction. Once the DNA extraction completed, we have to do a DNA fragmentation, which is around 300 to 400 base pair, since NGS is short read sequencer. Like it can sequence up to, it have a chemistry, different chemistry, 150 cross 2 and 300 cross 2. Means it can sequence up to 150 base pair and or 300 base pair. So this DNA fragmentation can be done by enzymatic method or mechanical or chemical method. So once the fragmentation is completed, we have to take this fragmented DNA to the library preparation, where we add adapters, or it is called as barcode. Barcode means it is just an ID number of a particular sample, because we will pool we have to pool number of samples in one tube, and we will be taking this to the sequencer. So we have to add a barcode which can identify our sample after sequencing. So once the barcode attachment is done, we have to do a library PCR, like enrichment PCR, which is library enrichment PCR. Once this is completed, again we have to do a quality check using qubit, which will tell the concentration, and tape station, which will tell the uh, library integrity. Then it can be, then all the samples, like we can sequence hundreds of samples in one single run. So once we have to pool all the libraries in one tube, then load it in the sequencing cartridge, and this sequencing cartridge can be sent to instrument for sequencing. Where once the sequencing is completed, we will get the raw data, which is in FASTQ format. It initially we get it in a BCL2 format, which will be converted to FASTQ format. This FASTQ format will be used for quality check, followed by the quality trimming, like adapter trimming will be there. Then it can be sent to the respective downstream analysis. It can be a genome assembly for WGS. It can be a that is variant calling. It can be like gene expression, or it can be taxonomic classification.
Sir, sir, your voice is not audible, sir. Sir, please unmute, sir.
So my slide is visible.
Yes, sir. Yes, sir. It's visible.
Okay. I think I was in this slide, right?
No, sir. That graphical yeah, this
This
slide.
one.
This we have
Okay.
finished, sir. Please go to next slide, sir.
Sorry?
This we have finished, sir, and please go to next slide, sir.
Yeah.
Yeah, this one, sir. Thank you.
So there are different techniques in marine genomics. One is whole genome sequencing, metagenomics, transcriptomics, eDNA monitoring, and epigenetics. This whole genome sequencing, metagenomic, eDNA comes under one category of library preparation, where we have to do a DNA isolation, which is followed by DNA fragmentation, library preparation, and sequencing, then analysis. This all comes under one category. But in transcriptome, it is different. We have to use a RNA for, first we have to use a RNA as our template, then which will be converted to cDNA. That cDNA should be fragmented, followed by the library preparation and analysis. Then in epigenetics, we have to do a bisulfite conversion of the DNA, which will be followed by the DNA fragmentation, library preparation, and sequencing. So in WGS, we can do the complete genome. And yeah, then in metagenomics, we can study the microbial diversity of any particular soil or any environment. Then in transcriptome, we can do a gene expression study in response to the any environmental stress, or in response to any environmental stress. Then eDNA monitoring uses the environmental traces of the DNA for biodiversity monitoring. Then epigenetic, we can study how the gene is regulated to the particular environmental condition. Yeah, these are the application. I have three applications. One is marine biodiversity conservation. So in marine biodiversity conservation, here we can use like identification of the species using DNA barcoding. We can detect rare and cryptic species, or we can also detect the invasive species, and assessment of genetic diversity within the population, conservation planning for endangered marine organisms. So yeah, as I told earlier, we can do a identification of species using barcoding. Then detection of rare and cryptic species or invasive species. These invasive species are one will kill our native species, so it is important to identify the invasive species. This genomic technique can help us in identifying the invasive species very earlier. So genetic diversity within the population. So even the population is similar, they can have two different they can be differ at the genetic level. So outcome of the study: ocean-wide conservation genomics. This is the article which uses ocean-wide conservation genomics of blue whale suggests new northern hemisphere subspecies. So in this study, they have used they have sampled for 509 blue whale, and they have used genome-wide association studies, with which they were able to identify two different genetic population, like two genetically different population. Like even though it is a single blue whale population, they used a genetic method to differentiate by at the genetic level. So, and they named this at the subspecies level. One is Balanthera musculus musculus and Balanthera musculus sulphurus. So, sustainable fisheries and aquaculture. Here we can monitor fish stock and population success, and then selective breeding for improved growth and disease resistance, prevention of overfishing through genetic monitoring, and early detection of pathogen in aquaculture system. Then what are the benefit? This can increase productivity, sustainable resource management. So one classic example is fishery genomics of snapper along the West Australian coast. Here they have used genome-wide SNP analysis, which revealed three broad genetic stock of snapper along the Western Australia coast. It means even it is a single population, there can be genetic difference, which can be separated as three like individual genetic stock. Then marine genomics and climate change. Yeah, we know that climate changes is uh, affecting the marine environment. It is a new challenge for us. So yeah, ocean is getting warmer, and we also see changes like ocean acidification, salinity, and oxygen level. So this marine genomics can help us in identifying the response of the particular organism to these changes. So it will help us in predicting the species response to environmental stress. And this can support us in climate resilience conservation strategies. So as I told, this is my conclusion. Like marine genomics is the technique which we can use to identify species, and population genomic study, and we can use to study this for health assessment. Like health assessment mean individual any in a group of like population, in a population it can have a any population can have a healthy animal, and other population can have a unhealthy animal. So this can be identified using genomic techniques, and this healthy population can be introduced to the unhealthy population for uh, conservation related aspect. So thank you for exploring the marine genomics with me today. And any questions?
Thank you, sir, for your wonderful and informative presentations. Participants, if you have any doubts, please post it in the chat box. So one question is there, sir.
1 second.
What is the meaning of aliens SPP.
What is the meaning of alien species? Is invasive species. Like every region will have its own native species. Invasive species means any foreign organism which will be introduced in the particular environment. Like in our country, we have tilapia and then catfish, which we can see in every lake. This catfish we are feeding those catfish, and this catfish is occupying our native fish population. This can be a predator. It won't allow the native fish to grow. Then, what is the use of
Another,
selective breeding in marine fisheries? So, like we are not doing it. This work is going and related to selective breeding occurs in aquaculture industry, and in marine means like selective breeding means we can introduce as I told, like we can introduce a healthy animals. These healthy animals we can identify using any genetic trait which has specific genetic trait, that is through S&P analysis. These healthy animals can be introduced to the like particular population. That will help in further growth of the healthy population in particular region. So that is called as genetic rescue. Healthy animals will be introduced to the unhealthy population. Yes, thank you.
Thank you to all the participants for your interesting questions. And thank you, sir, for taking time to address them.
Yeah, thank you, Sumesh, and sorry for the interruption. There may be some network problem. That was it. My PPT is not
It's okay, sir. It's okay.
continuously working. Sorry.
It's
Yeah.
okay, sir. Before we conclude today's session, I would like to share a few important updates with all the participants. I have shared some useful information in the chat box for your reference. This includes the chart sheet and requisition form for consultancy services available through the Center for Ocean Research and National Facility for Coastal and Marine Research. I have also shared the details regarding admissions for MSc Marine Biology program at Satyabama Institute of Science and Technology. Those who are interested in pursuing higher studies in marine biology are requested to go through the admission details shared in the chat box. Additionally, the details of internship programs being conducted during the month of August in COR and NFCMR have also been shared. The students and participants who are interested in gaining hands-on exposure and practical experience in marine science and related areas may kindly go through those details. I request everyone to take a moment to check the chat box and make use of these opportunities. Once again, I thank our speaker, Mr. Raghavendra Kumar sir, for the excellent and informative section. And I thank all the participants for your active participation. We look forward to welcome you again in the upcoming sessions of the Ocean Research Knowledge Series 2026. Have a wonderful day ahead. Thank you.